Hello, everybody. My name is Javier Martin. I am MD, PhD. I'm currently a professor at the Biomedicine Institute located in Granada, south of Spain, that belongs to the Spanish National Research Council. The scientific interest of our group in the last year was focused on the genetic basis of autoimmune diseases, now immune-mediated inflammatory diseases, mainly those with a rheumatic component, with a special focus in the last 10 years on systemic sclerosis. As you know, scleroderma or systemic sclerosis is a complex connective tissue disease of unknown etiology, but we know that genetic factors play an important role in the onset and development of the disease. A milestone in the genomics of scleroderma was the publication by our group of the largest GWAS, genome-wide association study of GWAS in the disease that included more than 9,000 individuals from several countries in Europe, the States and Australia, and we were able to identify around 30 independent risk loci. The first lesson that we learned about the genomics of the disease, thanks to this GWAS study, was that immunological alteration should be considered an early and central event in the development of the disease. So this was some debate in the field. What is first in the scleroderma? I think now we know that thanks to our genomic studies that immunologic abnormalities are first, later vasculopathy, and later fibrosis. One of thanks to our GWAS study, we were able to develop a polygenic risk score that can discriminate between scleroderma patient and control, and also other related systemic autoimmune diseases. This is a potential application clinical application to the genetic finding. That is a challenge in this field, that how to translate from gene to phenotype. And an additional potential application of genetic finding is the use of this genetic risk factor for drug repurposing. We know that in our GWAS we found several loci that are shared between different several mediated immune diseases related as rheumatoid arthritis. So finding shared genetic risk factor between these diseases can help us to identify a new drug for this orphan disease in terms of treatment, that is scleroderma. And finally, the great challenge for the future in the field is to know what is the functional relevance of the genetic risk variant associated with the disease, mainly because they lie in non-coding variants. So our group have developed and used different scientific approach to try to better understand what is the functional consequences of these genetic variants in the disease. That this approach goes from eQTL analysis, methylation analysis, and gene editing techniques. So thanks. I hope you will enjoy my talk.